Well, it's really a pleasure. Can you hear me okay? Is this one? Okay, it's really a pleasure to be here. And I'm grateful to all of you for coming out. It's really gratifying to see the turnout for this group of presentations, so thank you. But I'm also very aware that I am the only thing standing between you and lunch, so try and keep it moving and keep it interesting. So autism really has been front page news over the last 20 years or so. That's not always been the case, but I wanted to just kind of orient folks to this. We're taking a little bit of a switch, and this is a disorder that, despite all of our best efforts, really is a considerable challenge for families, for affected individuals, communities, and it's a disorder that affects children from the youngest ages throughout the lifespan. Now, over the last 10 years or so, there's been this new paradigm that I'll be talking about, and I think Ami will as well in the talk after lunch, and that allows us to focus on infants or babies at risk for autism. And this, I think, really has been one of the greatest steps forward in our field in allowing us to begin to see the early emergence of this condition. So really, prior to this line of research, I would say that the absolute vast majority, 99.9% of studies, have been done after people cross this line, or babies cross this line, and get a diagnosis. So a diagnosis that occurs at 2 or 3 or often later into childhood. But there's this period before then, and really we sort of skipped over that until recently. But what we now know, we've known this actually for a long time, that those babies that have older siblings with autism are at increased risk for having autism and really have a 20-fold increased risk. And so that has allowed us to go back and study these individuals more efficiently during this period of time. But the other piece of this story, I think, that's been extremely important in helping us to get a lot of traction in this area is illustrated by this work by Sally Osinoff, one of our colleagues. And this takes a very standard paradigm to look at reciprocal social interaction, in this case gaze to faces between an infant and mother. This red line are those children that end up developing autism. This dashed line are those kids with typical development. And what we see with the defining features of autism is that as early as 6 months of age, we really see no difference in these real demonstrable observations about social deficits in autism. But we also see this decline over time as these symptoms emerge. So essentially what we're seeing here in this first year of life is a period that we think of as pre-symptomatic for the defining features of autism or prodromal, much in the same way that we have come to think about schizophrenia over a long period of time since we've known that information. So now, this period of time in the first year of life where the symptoms of autism are first emerging is also a period of time of massive change in the brain. So I just use this kind of crudely to illustrate. This is work from John Gilmore's lab showing that between 2 weeks of age and 1 year of age, the brain is doubling in size. So you can imagine all the other more fine-grained changes that are going on during this period of massive change. And so when these sort of lines of work came together in our own group and in the community of researchers studying the behavioral emergence of autism, we put together a network, a national network that actually has collaborators throughout North America to collect a large number of babies at high familial risk for autism and examine them, in this case at 6, 12, and 24 months of age, with behavioral assessments and brain imaging, and other studies that we've done with different breakdowns and more frequent assessments. We call this the Infant Brain Imaging Study Network, and so I stand here as the representative of that group, but really this is one of those that really truly takes a village to get this work done. So I've kind of altered these slides a little bit just to make them easier to see in this audience, but the data is really the same. And this is a measure of a metric that we use to measure the brain. That's really not so relevant. Diffusion tensor imaging, the organization of white matter fiber tracts or the neural circuits in the brain. And we look at children, the same child at 6 months of age, at 12 months of age, and 24 months of age with this technique. The red line are those high-risk siblings that end up with a diagnosis of autism at 24 months of age. And the blue line are those high-risk siblings who don't get a diagnosis of autism. And so really the first lesson that we've learned, that we keep learning over and over again, is really nicely illustrated by this work. The first thing is that these trajectories are different. The second is that we see brain changes as early as 6 months of age. So this is well before even the emergence of the defining features of autism. And the third important thing that really gets at the fact that this is a developmental disorder that we have always studied cross-sectionally is that this brain signature, if you will, changes over time. So if we had done the study at 6 months of age, we would have concluded that autism had a higher value of this fractional anisotropy. If we had looked at 12 months of age, we would have said that there was no difference. And if we'd looked at 24 months of age, we would have had a completely different conclusion that this was actually decreased. And so timing in thinking about the emergence of autism, and probably even beyond the emergence of autism, is a critically important area that we really haven't focused on in our studies of autism. Now, I'm going to talk about a sort of simple phenomenon of brain volume, but really these findings really we're finding in every aspect of the imaging work that we're doing, in the diffusion tensor imaging work, and studying volumes, and studying functional networks in the brain. And these are really age-specific, disorder-specific changes. So here we follow up on the finding that was around in the field for a number of years. I first reported increased brain volume in autism back in 1992, and others have found that the finding is much more robust in preschoolers at age 3 and age 4. And we were able to ask the question with this design of when does it occur, how does it happen, and what may be its significance. So this is a paper we published last year where we followed a group of kids that had scans at each age, 6, 12, and 24 months, and that's really important in a heterogeneous disorder, in a disorder that we know changes over time. And what we found was that there were no differences in overall brain volume at 6 months of age, no differences at 12 months of age, and really the growth rate shot up between 12 and 24 months of age such that there were significant differences at 24 months of age. And what we also saw was that this was tied directly to the emergence of social deficits that are seen in autism. So when we looked at individuals, those individuals, the change in their volume during that time had increases in their social deficits. Now, in a very simplistic way, we sort of decompose volumes into this scenario, the thickness and the surface area of the volume, as you can see sort of here. And the surface area really represents the folds and the sulci and the gyri on the brain. And it's really what's responsible for the mammalian brain getting much larger. It's the increase in surface area. And we looked at surface area in this sample, and what we found was that it was the surface area in the first year of life that was showing a hyperexpansion or a dramatic increase in growth rate. We didn't see changes in cortical thickness, and it was, okay. We didn't see changes in cortical thickness, and it was really happening at this point in time. Now, just to remind you, that's a period of time that we call pre-symptomatic. As Ami will show you, there's also a lot of stuff going on at that time. but they aren't really the defining features of autism. We see problems with visual orienting and visual attention, problems with motor development, problems with sensory visual reception. And there's a well-documented story that underlies this expansion of surface area, showing that it's these immature or neural progenitor cells that are really driving this. And we also see, as expected, that this surface area change in the first year of life is directly related to this expansion of volume or increased growth rate in the second year of life. So when you put this together, it's just one example, I think, of a story that's happening in a way, I think a relatively new way to think of autism, is that it's not just the timing of what's going on early in the first 2 years of life, but it's really the sequence of events, and that there's this cascading effect of processes that we're seeing that really end up resulting in the emergence of autism. So we can start earlier, we can start, I think I'll leave that thought. I just got the sign. I think I'll move on. I made my point. So now we've made this observation that we're able to see brain changes early in the first year of life. But the diagnosis of autism has been later, as I've told you. There's been efforts to move this earlier. We've tried to use behavior in 18 months in this case. This may be the best study looking at typical autistic behaviors that really gave us sort of a 50-50 prediction of who was going to get autism as late as 18 months of age. So we went and looked at some of the different brain measures that we were seeing early on. And just to kind of cut to the chase on this, in the first paper that we published using a kind of a data-driven machine learning approach, looking at data from a scan at 6 months and 12 months of age, so within the first year of life, we were able to accurately predict 8 out of 10 kids that would get a diagnosis of autism by 24 months of age. This is using structural data. We did it with a different approach using functional data and also found very high accuracy of prediction. And we now have a third paper that we're getting ready to submit with a completely different approach, looking at 6 months data with a structural data that has an even higher prediction. So we're out here clinically diagnosing and treating autism, but the whole, I think, interest of our group is moving earlier. Earlier is better. We know that from medicine. We know that treating hypertension is better than treating stroke. We have evidence that earlier treatment of children once they have autism is better. We don't really know about pre-symptomatic treatment, but that's really the idea here. We know this is a period of that the brain is more malleable, that the symptoms and the brain changes have not consolidated. We haven't replicated this. We're getting ready to start a replication study after the first of the year, but we take this fact that we have 3 different approaches that are pointing us towards brain changes in the first year of life and as early as 6 months of age, accurately predicting later diagnosis as being really a proof of principle that we can get this done. This, of course, is in a group that's already been identified as high risk. So we want to replicate this, and we think that's an important group to study. There are a lot of families out there that have an autistic child and have a newborn. The ultimate goal is to try and move this into the population with other early, other kinds of predictors. You heard earlier something about polygenic risk scores and maybe some other behavioral measures, but to try and look at this in the population. I think I'm going to skip this work. How much time do we have? We have a couple minutes. All right. You'll have to hold off on the video. Just to summarize, understanding infant brain development is critical to understanding how to intervene before symptoms appear. Pre-symptomatic detection is possible, and we think that pre-symptomatic intervention may transform outcome. in this very challenging condition. Can't really read this, unfortunately, when the slides got reformatted. I didn't pay attention to this. But this is a group of about 30 or 40 people in North America that are all contributing heavily to this work. And I'm just really their representative today. So many thanks to them. Acknowledgments to our funding agencies. And I really want to underscore thanking the families. This is not a trivial contribution to these studies. They often fly to one of our sites for 2 or 3 days with their older child with autism and with their 6-month-old. They stay for several days and they come back 2 or 3 more times. So a lot of thanks to the families as well. Thank you.